Dear students, welcome to EPG Pathshala. I am Dr. Y.S. Rajput, former Head of Division, Animal Biochemistry Division, National Dairy Research Institute, Karnal. Today, I am going to teach you one of the important technique, polymerase chain reaction under the paper Biochemical Techniques. Polymerase chain reaction, in short, is called as PCR. Short name of the technique, that is PCR, is very popular. The nomenclature of the technique itself provide useful information. Polymerase word refers to polymerase enzyme. Chain reaction refers to reaction where product of one reaction acts as substrate of new reaction, and product of this reaction again acts as substrate for another new reaction. This technique is recognized as revolutionary technique for which inventor Carey B. Mullis was awarded with the prestigious Nobel Prize in 1993 for chemistry along with Michael Smith. Genes are segments of DNA, that is deoxyribonucleic acid, and DNA is polymer of nucleic acid bases. These are four different bases, namely adenine, guanine, thymine, and cytosine. Sequence of these bases in DNA and its size decides the characteristics of living cell or tissue or organism. There are about 25,000 genes in human body. Segment of DNA is required for numerous purposes, and it should be available in sufficient quantity and in purified form. One possible approach could be to isolate DNA, cut the DNA to obtain desired segment, and finally purify it. In view of the large size of DNA, this approach cannot provide the targeted segment of DNA in purified form. Carey Mullis conceptualized of a machine through which from a single molecule of DNA one can get multiple copies of at the end like a Xerox machine. In 1983, Carey Mullis developed the polymerase chain reaction, which is now most generalized technique used in any medical or biological science research labs for a range of applications. The technique is routinely used for gene expression studies, DNA cloning, phylogeny, functional analysis of genes, diagnosis of disease, and in paternity test. In current presentation, our objective will be to understand the principle of polymerase chain reaction. How this technique is performed will also be explained in today's presentation. Errors on bench and their solutions will be also explained while performing the PCR technique. The PCR technique can be performed in different designs. These designs in the form of variants will also be discussed today. Finally, I will be also explaining the different applications of the polymerase chain reaction. Dear students, so far we have covered the basic concept of polymerase chain reaction. What are the different steps in PCR are also covered. PCR is a powerful technique because millions of copies of target DNA can be synthesized from one PCR reaction. While doing it, specificity is maintained, and this is the beauty of PCR. Since its invention, several variants of PCR are created. Applications of PCR are now in diverse fields. These will be explained now. Polymerase chain reaction was conceptualized by Carey Mullis in year 1983. He was working with Cetus Corporation. The idea of PCR reaction came to his mind when he was driving US route number 101 in Northern California. In year 1985, PCR was introduced to scientific community. Cetus Corporation awarded Carey Mullis with $10,000 bonus for this invention. Cetus Corporation sold the patent on PCR to pharmaceutical company Hoffman LaRoche for $300 million. Polymerase chain reaction or PCR can be defined as laboratory technique employed for making numerous copies of segment of DNA. PCR is very precise and can be used to amplify or copy a specific DNA target from a mixture of DNA molecules. The principle behind polymerase chain reaction is based on ability of DNA to replicate into new strand of DNA complementary to previous template strand. Basically, it differs from replication of DNA in the sense that only a fragment of DNA is multiplied several times to get a multiple copy of the fragment. Polymerase chain reaction or PCR has become the most generalized technique used in biological science research labs for a range of applications. It amplifies double-stranded DNA molecule to obtain multiple copies of the same, and the sequence is also specific. The technique is simple, powerful, sensitive, specific, reliable, and very fast. We can get millions of copies of a single molecule of a specific sequence within 30 cycles of reaction. PCR reaction involves three basic steps. First one is denaturation of template DNA, which is achieved by heating DNA sample to 95 degrees centigrade. Second step is called annealing, which is at temperature 50 to 60 degrees centigrade. In this step, primers are annealed to denatured strands of DNA. Final step is called elongation step, wherein strand complementary to template strand is extended. These three steps are followed in sequence and constitutes one cycle. It is again repeated that in each cycle, reaction will undergo denaturation, annealing, and elongation steps. Denaturation step is the first regular step of PCR reaction and is consists of heating the reaction mixture containing the template DNA to a temperature around 94 degree to 98 degree for 20 to 30 seconds. This results in breakage of hydrogen bonds between complementary bases between adenine and thymine and between guanine and cytosine. This leads to denaturation of DNA. Consequently, DNA is unzipped to open and single-stranded DNA molecules are obtained. The time of denaturation should be kept optimum such that the complete portion of DNA strand to be amplified should be unzipped. Denaturation of DNA can be detected by increase in ultraviolet absorbance at 260 nanometer. This phenomenon of increase in absorbance is called hyperchemicity. In the next step, gradually the temperature of reaction is lowered down to around 50 to 65 degrees centigrade for 15 to 60 seconds, which allows the DNA molecules to re-anneal, but during this step, short stretches of single-stranded oligonucleotides called primers added in the reaction mixture incorporates and get annealed to single-stranded DNA template. The temperature selected for this step must be low enough to allow for hybridization of the primer to the strand, but should be higher enough to let the binding be sequence specific between template and primer. In other words, primers can only bind to a perfectly complementary sequence of the template. If temperature is selected very low, primers would bind non-specifically. When annealing temperature is kept too high, primers will fail to anneal as hydrogen bonds cannot form. In general, the annealing temperature is adjusted about 3 to 5 degrees below the melting temperature, that is the Tm of the primers. Tm is the temperature at which 50% of the oligonucleotides and its perfect complement are in duplex. It is dependent on length of DNA molecule and its specific nucleotide sequence. Theoretically, Tm value can be calculated from the number of all the four bases, that is guanine, cytosine, adenine, thymine, as per the formula where Tm is equal to 4 into the sum total of G plus C plus 2 into sum total of A plus T. Now the polymerase can bind to primer template hybrid and further starts formation of new strand complementary to the template. This is the step where polymerase attached to primer template hybrid starts extending the new strands of DNA by adding specific nucleotides, that is DNTPs, which are complementary to template strand into developing strands. So that's why this step is called elongation step. The temperature selected is different for different DNA polymerase. Most commonly used polymerase is Taq polymerase, which is derived from a thermostable bacterium, that is Thermus aquaticus, and has its optimum activity at temperature from 75 to 80 degrees centigrade. The time required for this step depends on both on the length of the target DNA to be amplified and on the efficiency of DNA polymerase used. In general, DNA polymerase can add 1000 bases per minute. At each extension step, the amount of DNA target is doubled. In PCR, the products of the first reaction becomes substrate of the following one, and so on. The three steps, denaturation, annealing, and extension, are repeated in a cyclic process, which gives this technique the name polymerase chain reaction, which means a chain of reaction involving polymerase. PCR is carried out for 25 to 30 cycles in an automatic thermal cycler, which can heat and cool the reaction mixture speedily. At the end of PCR reaction is given a hold at 4 degrees centigrade to stop the reaction and save the PCR product from degradation. In every cycle, product is doubled. It is theoretically calculated that single molecule of DNA can give 1 billion copies of template after 30 cycles of PCR. Dear students, now it should be clear to you that PCR technique involves 3 distinct steps, that is denaturation, annealing, and elongation. These 3 steps constitutes one cycle. Product of one cycle is used as substrate for the next cycle. In each cycle, product is doubled. Amplification uses polymerase enzyme and therefore reaction is specific. Polymerase enzyme requires oligonucleotide, which is complementary to the ends of target DNA and are from 5' to 3' direction. These are called primers. Primers must anneal to target DNA for polymerase action. Denaturation temperature, annealing temperature, and elongation temperatures are different. Thus, for cyclic reaction, reaction mix is brought to desired temperature for next step quickly. For quick cooling and heating of reaction mixture in low volumes in plastic tubes, machines are available, and these are called thermocycler or PCR machines. Let us know what are the various requirements for the PCR. We require DNA template, we require DNA polymerase, we also require buffer, deoxy NTPs, primers, dispensers, and thermal cycler. DNA template is the sample DNA that contains the target sequence to be amplified in PCR reaction. It can be genomic DNA, it can be plasmid, or even it could be cDNA. At the beginning of reaction, a high temperature is applied to this template DNA to separate the strand, called initial denaturation. For PCR, a very small quantity of DNA is required, as more amount of DNA template will increase the amount of contaminants and reduce the efficiency of polymerase chain reaction. DNA polymerase is the enzyme responsible for copying the sequence from single-strand DNA. The enzyme adds one base at a time at 3' end of primer. These enzymes should have capability to generate new strands of DNA by using the DNA template and primers. The enzyme should be resistant to high temperature. The enzyme should be thermotolerant. That is, enzyme must survive a temperature of 95 degrees centigrade, which is the melting temperature of denaturation step. PCR is cyclic process, and in each cycle, reaction mix temperature reaches 95 degrees centigrade in denaturation step. Primer annealing is more specific at higher temperature. Thus, thermostable enzyme will not be inactivated during annealing. The most common enzyme used in PCR is Taq DNA polymerase. It is isolated from Thermus aquaticus. It lacks 3' to 5' proofreading activity and results in low replication fidelity. It commits one error in adding 9,000 nucleotides. Other polymerase, PFU, DNA polymerase, which is obtained from Pyrococcus frueus, is widely used due to its higher fidelity when copying DNA. In addition, Taq polymerase also adds an extra adenine at 3' end. These extra adenine are useful for TA cloning. For any enzymatic reaction, an optimum reaction condition is needed, which is provided by the buffer solution. For example, buffer used for PCR reaction provides a suitable chemical environment for optimum activity and stability of the DNA polymerase. PCR buffer contains trace HCl, KCl or magnesium chloride and glycerol or tritone X100. In most cases, the PCR reaction buffer is provided along with the DNA polymerase and in 10x concentration, which is diluted to 1x in final reaction volume. High magnesium chloride concentration will give more product but low specificity. Low magnesium chloride will result in less product but specificity will be high. Deoxynucleoside triphosphate is the basic substrate for PCR reaction, which is a mixture of deoxy-ATP, deoxy-TTP, deoxy-GTP, and deoxy-CTP. Excess of deoxynucleoside triphosphate can increase rate of error and can also inhibit the activity of Taq polymerase. Lower amount of DNTPs may reduce rate of reaction. Thus, an optimum concentration of DNTPs is required for PCR. Usually, DNTPs are used at 10 to 50 micromolar concentrations. If we need to amplify large size of DNA, higher concentration of DNTPs is required. Primers are very important for PCR. These are short pieces of single-stranded DNA of about 20 to 30 nucleotides that are complementary to target sequence. DNA polymerase starts synthesizing new strand from the end of primer. Up to 3 micromolar of primers concentration is sufficient. Higher primer to template ratio may result in non-specific amplification and formation of dimers of primers. In most of the PCR, a set of two primers, that is forward primer and reverse primer, is used to amplify a specific region of gene. But a number of PCR have been designed which can run with a single primer, as in case of rapid amplification of polymorphic DNA, or in short referred as RAPD, where primer binds to several polymorphic sites in genome and amplifies all of these regions, resulting into PCR product of variable size. RAPD is successfully used for differentiating plant varieties, animal breeds, and microbial strains. While designing any primer, few points are to be considered. It should be of a size around 20 to 30 nucleotide. It should avoid formation of primer dimer. The GC content of the primer should be between 40 to 60%. The TM value should be between 55 to 65 degree centigrade. Increasing number of information about DNA sequences over specific various public database has made it easy to design specific primer sets. A number of free online bioinformatics tools can be used for designing primers and PCR conditions as primer 3, primer blast. The thermal cycler is the machine which heats and cools the reaction tubes to provide temperatures required for each PCR step of the reaction, that is denaturation, annealing, and extension. Most of the modern thermal cyclers make use of Peltier chips, which permits both heating and cooling of the block simply by reversing the electric current. Several thermal cyclers are equipped with multiple blocks, allowing several different PCR reactions to be carried out simultaneously. Some models also have a gradient function to allow for different temperatures in different parts of the block. This is particularly useful when testing suitable annealing temperature for PCR primers. Thermal cycler is available from number of companies including Applied Biosystems, Biorad, and Eppendorf. Any PCR reaction is most commonly carried out in 200 to 500 microliter reaction tubes covering reaction volume of 10 to 200 microliter in thermal cycler. Thin wall of reaction tubes permit effective thermal conductivity to allow for rapid thermal equilibration. Thermal cycler has heated lids which prevents condensation at top of the reaction tube. All the ingredients of PCR are mixed in this tube and the PCR is performed in a thermal cycler. The machine heats and cools reaction tubes as it is programmed to provide the temperature required for each PCR step of the reaction, that is denaturation, annealing, and extension. At the end of PCR, the reaction is given a hold at 4 degrees to stop the reaction and save the PCR product from degradation. After each PCR reaction, the result is visualized on agarogel electrophoresis of the PCR product, which should show a band of specific size for positive result. Absence of band or band of different size shows non-specific result. In this slide, the optimization of PCR reaction is described. Optimization of PCR is essential to achieve complete reaction in each step of each cycle. If elongation step results in complete and incomplete extension of primers, it will result in the synthesis of undesired products of variable sizes. The yield of the desired product, which is derived from completion of strand in extension step, will be low, and at times yield could be too low and will require performing PCR again. Optimization is done with respect to concentration of template, concentration of primer, concentration of magnesium ion concentration, and annealing temperature. Low annealing temperature results in mispairing, while the higher annealing temperature may lead to poor binding of primer to template. When non-specific products persist in spite of all attempted variations in reaction condition, nested PCR can be attempted. In this approach, PCR is carried out in 2 rounds. In first round of PCR, primers are designed to give product which must have a target DNA fragment. In second round, specific primers are used to amplify regions of target DNA. There could be troubleshootings, so it should be checked for DNA quality, and one can reduce the annealing temperature, one can increase the magnesium ion concentration, or use different thermostable enzymes. Throw out the primers if there is no reaction product. It is always advisable to make new stacks of the primers. PCR has got tremendous applications ranging from classification of organisms to disease diagnosis. The technique is used for classification of organisms in genotyping, in molecular archaeology, in mutagenesis studies, in mutation detection, in sequencing of DNA, in cancer research, in detection of pathogens, in DNA fingerprinting, in drug delivery, in genetic matching, in genetic engineering, and in prenatal diagnosis. PCR has been used in medical labs for genetic testing. PCR can be used for confirmation of genetic diseases at prenatal stage. PCR can be used for testing various types of cancer to study genetic mutation. PCR can also be used for replacing test for tissue typing for organ transplant. PCR can also be used for diagnosis of HIV disease. The method is more sensitive and easy than other antibody-based tests, where antibodies do not appear in bloodstream until weeks of infection. PCR tests are able to detect some infectious disease such as tuberculosis, malaria. This can be also used for sex determination of unborn child. Genetic fingerprinting can be a tool to differentiate a single person from the entire population. Very small amount of sample collected from the crime spot, such as hair, nails, blood spots, and body fluid, can be processed for polymerase chain reaction, and criminal can be identified. Evidence from decade-old crime can be analyzed. PCR has been found effective in DNA paternity testing, where DNA of unidentified person or parents can be tested and compared with that from possible parents' siblings. Such tests can also be performed for confirmation of biological parents of any dispute at court. PCR has been used in many research areas of molecular biology. PCR allows amplification of DNA even when we have no idea about sequence of DNA to be amplified. PCR can provide surplus of pure DNA molecules for techniques like hybridization experiments, where starting DNA material is very less or available in low amount. PCR assists in DNA sequencing, where we can amplify a particular gene to be sequenced and extract the segment from unknown individual, or can generate single strand from region of interest of any genome. In gene cloning, PCR can extract segments of DNA from large genome for gene cloning and can be further inserted into host by means of vector. By a single set of primer specific to the vector, the clone fragment can further be extracted out from the host. In molecular archaeology, PCR is used for phylogenetic analysis of DNA from ancient sources such as bones, frozen tissues. DNA from these samples are subjected to PCR, and after amplification, these are analyzed for their phylogeny. We cannot imagine biological research lab not using PCR technique in their research program. PCR reactions are modified to generate its more wide applicability for different applications. So for that purpose, a number of variants are now available which helps in having the particular specific applications. With slight modification protocol of PCR, a new variant of PCR can be developed to get desired amplification. In this way, a variety of PCR have been developed, such as multiplex PCR, nested PCR, touchdown PCR, hot start PCR, and asymmetric PCR. In this slide, the concept of multiplex PCR is explained. In multiplex PCR, a number of different set of primers are used in a single reaction where primers anneal different target sequences. Thus, it permits analysis of multiple targets in a single reaction. Multiplex PCR has been used for analysis of microsatellite DNA and single nucleotide polymorphism. I will be explaining another variant of PCR, that is the nested PCR. Nested PCR is used for increasing the specificity of PCR. In this, two sets of primers are applied in two successive reactions. In the first reaction, one primer set generated a product which contains regions from non-target area. In the next reaction, product from first PCR is used as a template, and one or both the different primers whose binding sites are present within the first set, which increases the specificity of reaction. It has been found more successful in amplification of large DNA products than normal PCR. So this technique is specifically used when specific amplification of the target DNA is required. This is another variant of the PCR referred as touchdown PCR. Touchdown PCR is also a variant of PCR in which the annealing temperature of reaction is decreased gradually in later cycles. In early cycles, the annealing temperature is programmed usually 3 to 5 degrees above the standard melting temperature of the primers, while in the later cycles it is kept below the Tm value. Higher initial temperature provides greater specificity for binding of primers. Lower temperature in later cycles helps in efficient amplification of DNA. In hot start PCR, this PCR is performed manually by heating the PCR reaction components to a temperature of 95 degrees centigrade before addition of DNA polymerase. It reduces the non-specific amplification of target DNA. The limitation in the PCR that it requires primers complementary to both termini of target DNA is taken care in inverse PCR. Because in inverse PCR, even if the target DNA sequence from the one particular region is available, then amplification can be carried out. By inverse PCR, a target region with an internal section of the known sequence and unknown flanking region can be identified. Dear students, let me summarize what we have discussed. PCR or polymerase chain reaction is a molecular tool used to amplify a sequence of DNA by using a pair of oligonucleotides, primers, each complementary to one end of the target DNA sequence. These two primers are extended towards each other by a thermostable DNA polymerase in a reaction cycle of 3 steps, that is denaturation, primer annealing, and polymerization, which is performed in a thermal cycler. Carey Mullis invented this reaction, and the reaction is now used in different formats so that it can be used for different applications. This reaction has left its permanent marks on molecular biology research. It has resolved many cases of crime and paternity dispute. The technique can be performed by technical person. Availability of the reagents and enzymes from several commercial houses has helped in its wide use. We cannot think of a molecular biology research laboratory not using the polymerase chain reaction in a routine way. Thank you.